Hello, this is 16.4. The glycolytic pathway is tightly controlled or regulated. So we're talking about glycolysis and the regulation of glycolysis. There's two main goals of glycolysis. Of course, one is to generate ATP. We know that net 2 ATPs are made per glucose. A second goal is to generate small molecules or building blocks for anabolic synthesis. That is, some of the glycolytic intermediates are pulled off to create small molecules from those that can be used for building macromolecules. For example, pyruvate can be changed into alanine, an amino acid, in case protein synthesis is needed. Other glycolytic intermediates can be pulled off to make different amino acids. Also, cells doing glycolysis might be in need of forming fatty acids, so they need carbons for making fatty acids or building blocks for making fatty acids. So two main goals we need to think of, and one is synthesis and one is energy, making energy and energy regulation. If we already have enough energy, we can slow down glycolysis. If we already have enough building blocks, we can slow down glycolysis. So reactions of glycolysis must be regulated to meet these two major cellular goals. 3 enzymes of glycolysis are regulated. These are the 3 irreversible enzymes, the ones with the big negative delta Gs. The first reaction often is the committed step and the regulated step of a pathway. In glycolysis, it turns out the third reaction is the committed step, not the first reaction. The third enzyme of glycolysis is phosphofructokinase, abbreviated PFK. It is the most important regulation step of glycolysis. Also, in glycolysis, the last reaction of the pathway, the last enzyme, pyruvate kinase, is regulated. Notice that these 3 are all kinases, and these are the only 3. Well, that's not true. There is another kinase in glycolysis, and it's not regulated. So we need to memorize these 3. Hexokinase, phosphofructokinase, and pyruvate kinase are the regulated sites of glycolysis. So remember, regulation in general can be done in 3 main ways. Transcriptional control, where a gene is transcribed and then translated into protein enzymes. Allosteric control, which you learned a lot about in Biochem 1. Allosteric control is where there's an enzyme that not only has a catalytic site, but it has a regulatory site as well. And small molecules can bind to the regulatory site to slow down the protein or speed up the protein, the enzyme protein. Also, covalent modification is a third way to regulate enzymes, and phosphorylation or dephosphorylation are very common covalent modifications that can slow down or speed up an enzyme. Notice that allosteric control is reversible in milliseconds. Phosphorylation and dephosphorylation can change the activity of an enzyme and can be reversed in seconds to minutes, whereas transcriptional control takes the longest, can be reversed in hours. Once you've got enough enzyme, your body will have to break down those enzymes or make new ones. Could take an hour or more. So allosteric, very fast. Phosphorylation, seconds to minutes. Transcriptional control, the slowest. Key point of regulation, PFK. This is the committed step. It's the most important regulatory enzyme of glycolysis. It's regulated differently in muscle than in liver. So this chapter is going to compare and contrast the regulation of glycolysis in muscle cells and in liver cells. So we need to think about what muscle cells do and what liver cells do. Muscle cells need ATP to do their contraction, to contract the muscles. They don't do synthesis. Liver, on the other hand, is all about synthesis. It synthesizes a lot of the molecules you need in your body, like cholesterol and others. Liver does not do anaerobic glycolysis, so it's not doing lactate fermentation. It's doing the opposite of it, actually. So it's not creating acid in the cell, so we don't need to worry about the pH being lowered. So the pH is not going to affect the rate of PFK, phosphorufructokinase, in the liver. Let's look at the right side of this slide. This shows a schematic diagram of PFK. Notice it's got 4 different subunits. Each of those subunits has a catalytic site. So there's 4 catalytic sites. Each of those subunits also has allosteric sites. So we are going to study each of the 3 enzymes, regulated enzymes of glycolysis, in the muscle and in the liver. So, wow, that's getting complicated. Luckily, we're only looking at muscle and liver. So regulation of PFK in muscle. I'm going to use the notepad and draw some diagrams here. So let's start with glucose. Okay, so remember, glucose is a one-way reaction catalyzed by hexokinase. And hexokinase is the main enzyme in the muscle, but in liver has only a little bit of hexokinase. It has a lot of glucokinase. And glucokinase is an isoenzyme of hexokinase. They're isozymes of each other. That means they have different amino acid sequence, which means they are created from 2 different genes. They both bind glucose. They both use ATP to phosphorylate glucose. They both produce glucose 6-phosphate. But they have different regulatory sites, and they're regulated differently. The hexokinase is going to have feedback inhibition by the glucose 6-phosphate. So glucose 6-phosphate inhibits hexokinase, whereas glucokinase is not affected by glucose 6-phosphate. Instead, it's only inhibited by phosphorylation. So hexokinase has an allosteric inhibitor. Glucokinase is changed by covalent modification by phosphorylation. Okay, so glucose 6-phosphate. Oh, I have to stop. So this is the end of part 1, and I'll continue this in part 2.